Hi everyone, my name is Emma Sewell and I am a biology student here at Florida Gulf Coast University. I'm going to be presenting on the dopamine release in the nucleus accumbens of rats based upon the intake of either pure hydrocodone or Vicodin. I'm going to be giving just a brief overview of the key points in each section, so let's go ahead and get started. The main focus of my project is to compare Vicodin and hydrocodone, which are two known addictive narcotics. Both of these drugs are commonly prescribed to patients with severe pain. Vicodin is the combination of pure hydrocodone and acetaminophen, where 97% of the drug is acetaminophen and the other 3% is hydrocodone. Acetaminophen is found in Tylenol and is completely non-addictive. The other component of Vicodin is hydrocodone, which is highly addictive and classified as a Schedule II drug by the FDA. Schedule II drugs have a high potential for abuse. Overall, though, Vicodin has a lower potential for abuse as it is labeled Schedule III by the FDA. To test the addictive qualities of these two drugs, I need to measure their dopamine release in the brain. Dopamine is a signal from the reward center of the brain in response to pleasurable experiences like drug use. With this information known, I want to sample the dopamine release in an area known as the nucleus accumbens of the brain through a microdialysis technique to understand why Vicodin has similar addictive qualities as pure hydrocodone, instead of being more closely associated to a non-narcotic like acetaminophen. So in other words, why is Vicodin 97% acetaminophen, but related more to pure hydrocodone? So here I just have the two chemical structures, one for Vicodin, one for pure hydrocodone. As you can see, Vicodin and pure hydrocodone have similar structures, but Vicodin has the addition of that acetaminophen group, and the molecular weights do differ, with Vicodin obviously being heavier. There are various key players in my proposal, so I want to briefly go through each idea. To start, this is an image of the location of the nucleus accumbens in the brain. It is shown in red in this image, and it is situated right between the putamen and the caudate. The nucleus accumbens is most widely recognized for its role in the reward center of the brain. It is a component of the ventral forebrain, and it ultimately acts as this liaison between the dopaminergic pathway and the central gray nuclei. The dopaminergic pathway is sometimes called the dopamine pathway, as it controls the neurons which release the dopamine neurotransmitter. Dopamine plays a large role in how we feel pleasure and how we receive reward. So for example, when you ingest an opioid, neurons from the ventral forebrain project onto the nucleus accumbens, which causes the shell component to quickly release dopamine. Not all drugs spark equivalent concentrations of dopamine release. Concentration release is dependent upon the addictive qualities of the drug. Both hydrocodone and Vicodin trend toward higher ranges of extracellular dopamine release. However, it can be hypothesized that Vicodin should release slightly lower or even significantly lower levels of dopamine concentration because it does contain that 97% non-addictive acetaminophen. This leads right into my research objectives. So I want to investigate the effects of Vicodin and hydrocodone on rats to ascertain whether pure hydrocodone or Vicodin releases significantly less dopamine in the nucleus accumbens. This would show which drug would not stimulate the reward center of the brain and thereby be less addictive by nature. To measure dopamine release, a microdialysis probe must be inserted into the nucleus accumbens of the brain. As you can see in the illustration below, a syringe pump as well as an outflow pump will be attached to the probe. The syringe pump acts as the inflow, which will be filled with perfusate, and perfusate will push the fluid out of the nucleus accumbens so it can be collected. The outflow will contain the dopamine, which will be stored in vials. These vials will then be ran through high-performance liquid chromatography to quantify the exact percentage of dopamine released. There are 4 treatment groups, as you can see. It's going to be pure hydrocodone, Vicodin, acetaminophen, and a control placebo. So acetaminophen is, like I said, completely non-addictive, as well as the control. So that would be an interesting comparison. But my main comparison is really looking into pure hydrocodone versus Vicodin release of dopamine. The experiment will be repeated 3 times for experimental validity, and I will use the analysis of variance test to see the significant differences between the 4 groups. The average will then be taken, and the average between all 3 trials would be recorded also. As we all know, drug addiction is a prevalent disorder in the world today. There are over 19.7 million American adults battling with addiction. With that being said, it is very, very important to find specific drug alternatives for pain. This can possibly be accomplished by analyzing dopamine release in the nucleus accumbens and seeing which drugs release increased dopamine levels and which do not. The drugs which limit dopamine release also lessen the chances for addiction. In this study, I specifically looked into 2 addictive narcotics prescribed worldwide, and both have the power to limit severe pain. So the ultimate question would be, which drug should be prescribed more in hopes of reducing the dependency on addictive drugs worldwide? Many researchers around the world study addiction science and work every day to answer these types of questions. I'm hopeful for answers in the future and look forward to learning more about the brain's response to addiction. Here are my references, and thanks for listening, everyone.